to another edition of Global Nerve Advancements. My name is Ann Engemann, and I am a member of the Global Nerve Foundation Research Committee. And today I will be talking to Dr. Damiano Barone, a neurosurgeon at Houston Methodist who specializes in complex peripheral nerve surgery. Today, he's here to tell us about his publication on functional neurological restoration of amputated peripheral nerve using Biohybrid regenerative bioelectronics. Welcome, Dr. Barone.
Thank you. It's a pleasure to present today the work on functional neurological restoration of amputated peripheral nerve using Biohybrid regenerative bioelectronics. The beauty of the nervous system is that everything is connected and wired. A signal starts from the brain, travels down the spinal cord and enters the nerve before finding its way to the muscle. Then muscle contracts in a coordinated fashion and generates a variety of movement which can go from waving a hand to playing the piano. Every year, there are between 1 and 2 million episodes of nerve injuries worldwide, resulting in many people living with long-term disability. While regeneration is effective in short distances, long-distance regeneration is unsuccessful. So what is our challenge? We must be able to bring the wealth of information that is still present in its entirety proximal to the injury, either in the brain, spinal cord or the nerve itself, and use it to achieve useful function. One way we can do was already suggested over 70 years ago by Seddon himself. He noticed that an amputation, a use of a passive prosthesis, will give the patient a more useful arm than any attempt to reconstruct the nerves in a flaying limb. Nowadays, we can do a bit better than with a passive prosthesis. We can use the residual muscle function proximal to the injury to control a neuroprosthetic device. Muscles generate stronger signal than nerves with higher amplitude and surface area, which can be recorded using simple electrodes. Another step forwards towards the objective to get as much information still present proximal to the injury is targeted muscle reinnervation, TMR. We connect proximal nerve stumps, originally meant for distal arm muscles, to larger muscles in the chest and shoulder. EMG electrodes then use these signals to control prosthetic arms. This strategy has also the added advantage as by giving the nerves a biological target, minimizes retrograde degeneration and neuroma formation. However, the movement achieved are still basic and surface electrodes become unreliable during vigorous movement and perspiration. To overcome some of these issues, one more step forward is the use of regenerative peripheral nerve interfaces, RPNI. Instead of using large muscles, small pieces of cut muscle about 1 centimeter in size are wrapped around the ends of amputated nerves. Electrodes are then implanted into the muscle pieces to record amplified signals. These signals can control prosthetic devices and provide a target for nerves, reducing both degeneration and pain. The main advantage of RPNI over TMR is that the electrode is implanted, eliminating the issues with movement and perspiration. However, the signal remains basic because each nerve, containing thousands of axons, is treated as a single signal rather than multiple distinct signals. So what are we missing? Current technological limitation force us to treat nerves as single signals, like an on-off switch. However, each nerve contains thousands of signals necessary for complex movements, such as typing or on a keyboard or playing the piano. If we can unlock this potential and record at a much more granular scale, robotic technology is ready to be utilized. The clear candidates for this advancement are neural interfaces directly connected to the nerve. Such neural interfaces need to record reliably from single axons, wherever they are implanted, last for the lifetime of the patient, and they should cause no harm. In our lab, the aim has been to design a novel approach that does not rely on external skin electrodes. We focus on maintaining the health of nerves and axons, preventing neuroma formation and reducing scarring by creating biomimetic devices. To achieve the best possible signal from each axon, we integrated what we learned so far into what we have called Biohybrid implants. Instead of using large muscles in the centimeter scale, we utilize muscle cells at the micron scale directly on the neural interface. As neural interface, we used our core technology in our lab, a thin film electrodes, which is only 4 micrometer thick, making it thinner than human hair. This electrode has the capacity to have a very high electrode density and conform to the difficult soft surface of a nerve stamp. For this project, we decided to use 32 electrodes in a 2 by 2 millimeter surface area just to provide proof of concept. We also needed cells which could be used off the shelf and rapidly populating at high density on our electrodes. We work with the Cotter Lab and BBio to use the OptiOx system. With this system, human pluripotent stem cells can be induced to become a pure population of myocytes, muscle cells, in less than 10 days. And you can see from this video how our myocytes differentiate in myotubes, which have the full contraction capability required to obtain an amplified signal. And this is our workflow. The device is manufactured and then sterilized, put on a petri dish, and cells are seeded on the implant and protected with fibrin glue and hydrogen. Once these human stem cells are fully differentiated to muscle cells, the device is ready for implantation. Here you can see a microscopic picture with the myotubes on our device, demonstrating a very good adherence to the electrodes. We implanted a Biohybrid neural interface in the nerve bundles of the forelimb of a rat, with the other side secured to the inside of the skin. One issue is to keep these cells alive, as they need constant supply of oxygen and macronutrients. Our hypothesis is that we could achieve this by suturing the device to the derma, which is highly vascularized tissue. The derma would then send small vessels by microholes in the device, which are less than 10 micron in diameter. First question: are the device actually conformable to the stamp, and have the cells remained in place? Here you can see the histology post-explant of the nerve and implant. On the top, the nerve itself, in the middle, the implant, and the bottom of the skin. You can appreciate how the device is deformed to exactly follow the nerve shape, demonstrating excellent adherence and conformability. Our next question is, do cells actually survive? Using a different stain, we're able to assess the survival of the stem cells that are myocytes. Because we use a xenograft, human cells in rat recipient, we can use human-specific stains to see if there are still human cells left. At high magnification at the bottom, you can see that human cells, the myocytes, are of a different color, a red-brown. So there has been no takeover from the host cells, and our cells have survived in a very good number. Our next question is, yes, they are where they're supposed to be, they survive chronically, but do they actually make functional connections? So we wanted to know if the nerve axons made contact with the muscle cells, creating new neuromuscular junctions, and remained alive. In green is the beta-3 tubulin, representing axon. In purple, acetylcholine receptor, pointing to neuromuscular junctions. As you can see, our Biohybrid models on the bottom has a significant increase compared to a device without cells on the top. You can also notice that top has a marked decrease in axons, which points to what we said before: an axon without a target degenerates. This is one of the most exciting results, as for the first time we show survival of transplanted stem cells, derived cells, and functional integration into the host tissue. Then we wanted to see if the signal at 4 weeks post-implantation was actually amplified in the Biohybrid implant more than in the electrode without cells. To have a consistent validation, we use hook electrodes to stimulate the nerve proximally and record these signals from our device. The Biohybrid was able to pick strong signals at a level where the normal electrodes couldn't. And just to clarify, the normal electrodes we're using are already the state-of-the-art among neural interfaces. We still were not happy and wanted to further prove this was not a false positive. So we carried chronic recording in the rats, and what we saw is what we wanted to see. No signal in the first 2 weeks, but only in week 3 and stronger in week 4, and this is because it takes 3 to 4 weeks for neuromuscular junctions to form. Lastly, we also wanted to see that the signal was not only random firing, but purpose-driven. With the limitation that these were amputated nerves with no moving limbs, we could still derive function from the task to be performed and from the contralateral limb actions. We observed discrete firing only when the animal was engaged in the task and nothing in between. And this was stronger in week 4 once there were more functional connection as expected. So in conclusion, we have come up with the concept of biohybrid neural implant merging two very different concepts. On one side, electrical interfacing, which provide the advantage to adapt and shape the implant to any anatomy and tissue and being able to actively control it. And the other side, cell transplantation, where cells can be used to integrate and interact with biological tissue. A biohybrid neural interface allows us and the patient to control the implanted cells, which integrates well in the tissue without excessive and deleterious scarring or tissue response, and achieve a chronic selectivity not currently possible with any neural interfaces. Thank you very much for your time. This is the paper with the QR code.
Well, thank you for that overview of this very interesting study. I'm curious, what advantage do you see for these biohybrid devices over techniques like targeted muscle reinnervation or regenerative peripheral nerve interfaces, which are some techniques for amputated nerve ends that have become quite popular in the last few years?
It's an excellent question, and I tried to cover some of this in the show presentation, but TMR and RPNI, excellent techniques, and they're useful both as a pain management tool, but also used as a neural interface and neuroprosthetic tool. The main issue is that TMR and RPNI, as I was saying, they treat the nerve as a single entity, so one nerve, one signal, while a nerve has multitude of signals. So every nerve has got thousands of axons, and thousands of axons is thousands of signals. So how do we extract this signal through, but without losing the advantage of having the RPNI and TMR, which is basically still giving a target for the nerve, so reducing that pain that comes from an amputated nerve. And that's what I think the biohybrid comes. Biohybrid is the vision is to basically having one axon, one muscle cells, one electrodes. So to truly take every single information from the nerve and transfer it in a neuroprosthetic. So that is the vision. That's what we're trying to do with this technology.
And my other question is what other applications do you see for these devices, or what applications are you most excited about?
This is a very much a platform technology. The idea is that we have now the ability to put cells and electrodes together. And then we can be very creative on the design of the electrodes, the type of the electrodes, and the type of cells. So in this case, we try to use, we say the most simple was very, very complex, but it was a simple model where we wanted axons inside the body to connect to our implanted muscle cells and create a functional connection, a neuromuscular junction, and then connect it to the electrodes. But if we think a bit more outside the box, then we can start playing with neurons itself. And rather than accepting a connection, we are sending connection. So we're now several projects in the lab where we're trying to experiment exactly that. Rather going in the proximal stump, going to the distal stump, for example, sending sensory or motor neurons, and we're doing things even in the spinal cord and the brain with different type of cells with different neurotransmitters. So the reality is that with this platform of technology really unlock a lot of possibilities where imagination becomes the limit.
Well, it definitely sounds like there's some very exciting research coming as you move forward with this.
It is. We just recently published another nice perspective review where we did a very nice review that, in 2019, trying to see what has been done in biohybrid field until that point. And I think this was the moment to try to revamp it. And we just published again in Advanced Materials. And it gives, again, another view, in particular in the last 5 years of where we've been, but in particularly trying to give our vision of where we are going. So if people are interested, it's called The Future of Biohybrid Electronics and has been published in Advanced Material last month.
Well, great. Thank you so much for taking time out of your busy schedule to talk to us today. It's been a real pleasure.
No problem. Thank you so much for having me.